Okay, good morning, everybody. Thank you for coming. It's a pleasure to introduce Larry Karnitz from the Mayo Clinic, where he is a professor of pharmacology and experimental therapeutics. And I invited Larry because I think he's a wonderful example of somebody who has equally successfully produced mechanistic basic research and translational science. So Larry is actually an alumni of the University of Minnesota, and he got his PhD at the University of Iowa, then went on to do postdoctoral research at the University of Washington in Seattle. And a little over 20 years ago, he started his own lab at Mayo. Sorry. And 20 years ago, not much was known about the DNA damage response and about DNA checkpoints. There was a little bit in yeast, but it was basically a black box in humans. And Larry's lab is credited with making a seminal contribution by discovering and identifying the homologs, human homologs of the yeast 911 checkpoint clamp. And he went on to show that 911 has a crucial role in activating the ATR check1 checkpoint pathway in response to replication stress. And over the years, he really has succeeded to translate this into clinical trials with checkpoint inhibitors in ovarian cancer. He has an impressive record of achievement. He currently holds 2 R01s, has a project on the ovarian cancer spore, and is a co-investigator on multiple other projects. And so I want to thank you for being here today and telling us about altering DNA damage responses to enhance chemotherapy outcomes.
There, and inviting me down today. Yeah, it was beautiful to see the sun and drive down or up from Rochester, and then hit the traffic jams, which is one thing we don't have in Rochester. So yeah, I would like to tell you some stories today about some of the research we've done that mixes both sort of basic research as well as ability to translate that research because of the environment that we're in. Basically, I'm going to tell you 2 stories. One, I'm going to tell you about a gene that's mutated in ovarian cancer that we've done a lot of work on, that when it's mutated actually confers defects in homologous recombination, and I'll come back to that in just a second. And then I'm also going to tell you another story that also is, we played out mostly in ovarian cancer that looks at base excision repair, which, as most of you probably know, is one of the oldest DNA repair pathways, and it's been extremely well studied, but we ran across a couple of new findings there that actually have led to an interesting clinical trial that's underway. So as long as we're going to talk mostly about ovarian cancer, I thought I would just give you the biology, medical aspect of this disease. The disease is often chemosensitive, but the 5-year survival rate is only about 30%. That's despite very aggressive surgery and chemotherapy. The disease is characterized by defects in homologous recombination DNA repair. Most of these are caused by mutations in BRCA1 and 2, which I'll tell you a little bit more about in a second, but they don't account for all the defects in the homologous recombination repair. Just to orient everybody about homologous recombination repair, it plays 2 roles. I'm only going to talk about one here. In one hand, it's very important in repairing double-strand breaks, but it also plays a role in restarting stalled replication forks, which actually probably becomes important later. But if you look there, you can see a double-strand break up on the upper left-hand side and the sister chromosome that's not broken, and we have a series of pathways that are activated, including the ATM kinase and the NBS complex shown below that. And those complexes process this double-strand break into long single-stranded regions that are then eventually loaded on, RAD51 is loaded onto those single-strand breaks, and that process requires BRCA1 and BRCA2. Hence, defects in those genes cause defects in this pathway. Rad51 filaments are then able to perform a homology search and recover the missing information from the sister chromatid, and then a whole bunch of resolvases take care of the final structures. Okay. So as I told you earlier, ovarian cancers are characterized, or at least high-grade ovarian cancers are characterized by mutations that disrupt homologous recombination. And back in 2011, the TCGA analysis was published that identified some of these mutations. And again, they found lots of mutations in BRCA1. Both these occur somatically, or germline as well as somatically. And they account for about 15 to 20% of the mutations. Some people call defects in homologous recombination a BRCA-ness, although that term's sort of falling out of favor. But we know that about 50% of ovarian cancers, perhaps even more than that, actually have defects. And many of these defects, or a good percentage of these defects, are not accounted for by mutations in BRCA1, BRCA2, or even some of the other homologous recombination repair pathways. So when this study came out, one of the things that caught our eye was, here's the list of genes that was most frequently mutated in the 316 samples that were analyzed, and you can see P53 is mutated almost universally. BRCA1 and 2 are mutated at a low level. It's a lower level in this particular set of samples because this is the somatic mutations, not the germline mutations. But one that caught our eye was this gene down here called CDK12, which was mutated almost as frequently as at least somatic mutations in BRCA1 and 2. And when we started looking at and learning about CDK12, there was very little known, actually. But its kinase domain is most closely resembled the kinase domain of CDK9. Okay, so these are CDKs. You might think they're involved in cell cycle regulation, but these CDKs are actually involved in regulation of gene expression. And unlike CDK9 and most other CDKs, CDK12 has a very long amino terminal extension and a C-terminal extension, so it's quite a large protein. If we look at what CDK9 actually does, it's involved in regulating gene transcription, and it phosphorylates RNA polymerase II on a tail that protrudes off the enzyme complex, and this tail has 52, at least in humans, has 52 repeats of a heptad sequence, and several of the serines in the heptad are phosphorylated by various CDKs that regulate transcription. And CDK9, what it does is it plays a role in phosphorylating these and converting a paused RNA polymerase complex that's initiated and sitting there on the gene, converting it into an elongating complex, and then traveling and finishing transcribing the gene. Now, at the same time that these TCGA studies came out, another group, which also caught our eye about CDK12, had done a study saying they were actually interested in cyclin K, and they started looking for the CDK that was associated with cyclin K. And the one that they found was CDK12, and so they did some studies, knocked down CDK12 and cyclin K in HeLa cells, and what they found was, when they did RNA-seq, is that a small number of genes, very small, about 0.5% were upregulated and about 2% were downregulated when they knocked this down. And when they looked at what genes those were, they found that many of them were involved in DNA repair. And in fact, when they did some Western blots, they could show that BRCA1 levels were reduced in those cells. And so when we saw that and started thinking about CDK12, we started asking questions, well, what are the mutations in ovarian cancer in CDK12? How do they affect its function? And so the mutations that show up when you look at the original TCGA data, there were a couple of frame shift mutations or premature stop codons in the amino terminus, and then there was a bunch of mutations within the catalytic domain. And so we asked some simple questions about this. The first one we asked is, how did the mutations that are in the catalytic domain, how do they affect catalytic activity? And then also, how does disrupting CDK12 function in ovarian cancer cells affect those cells? So, a graduate student in the lab set up a nice kinase assay for CDK12, which turned out to be a little bit more complicated than we anticipated. And so Purval Joshi did this, and what she did was made some expression constructs for CDK12 and then expression constructs for a bunch of the mutants. And the first two here are actually kinase dead mutants that mutate key lysines. One mutates a key lysine that's involved in catalysis, and the other one mutates the conserved DFG domain, both of which are required. So those were our negative controls. And then the remainder were the mutations that are found in ovarian cancer. And what you can see is that she used a RNA polymerase as a substrate. And then what you could see is that all these mutations, save one, are extremely kinase defective. And then that's just quantitated here. So she then went on to ask, well, what happens if I get rid of CDK12 in ovarian cancer cells? So she knocked it down with a couple siRNAs, and just like was seen in the HeLa cells, BRCA1 levels go way down. And given that BRCA1 levels go way down, our hypothesis was that there would be defects in homologous recombination. So to measure that, we did a couple things. One is we looked at the deposition of RAD51 onto DNA by measuring RAD51 foci. And you can see that when we knock down BRCA1, we have a profound defect. When we knock down CDK12, there's a defect, but granted not quite as large. And the other assay we used was actually to look at whether homologous recombination occurs. So we used a cell line where we had engineered into it a GFP that was broken, so that to get green fluorescence, you actually have to introduce a break, have recombination occur. And so using that assay, we could show that CDK12 depletion also caused defects in actual homologous recombination. Okay. So then the other question we asked was, well, if just to make sure that these kinase defective mutants were really defective in homologous recombination, we knocked down the endogenous CDK12 and put back either siRNA-resistant wild type, catalytically inactive, or just we just used one of the mutants in this case, and said, how does it affect RAD51 foci formation? And what we saw is that when we knocked it down, we could reduce RAD51 foci formation, putting back in wild type restored it, and putting back in the mutant did not restore it. So we could now show that not only does knocking it down, but expressing these kinase dead mutants, or these mutations that are found in ovarian cancer actually block homologous recombination. Yes?
Were these dominant negatives? That is, so you're putting it in the context of wild type?
We're getting rid of the wild type with siRNA, so we're putting in siRNA resistant. Yeah, you can see up here, blotting for CDK12, there's none here, and then we're restoring. Yeah, that's a good question of whether they're actually dominant or not. We've not looked. Nobody's done a really good analysis of what happens in tumors, whether they're both alleles. In some cases, both alleles are either mutated or one's missing, but they could have potentially dominant negative effects. So then, if these cells are really homologous recombination deficient, we would predict that they'd have some defects in their ability to withstand treatment with agents that cause intrastrand crosslinks, where homologous repair is really important, as well as defects or increased sensitivity to poly-ADP ribose polymerase inhibitors, which are sort of synthetically lethal when you have a defect in homologous recombination. We'll come back to that later. But what you can see is when we knock down CDK12 versus BRCA1, we have increased sensitivity to melphalan, which introduces intrastrand crosslinks, similarly to cisplatin. And then to the PARP inhibitor, veliparib, also known as ABT888, we had increased sensitivity that in this case was very similar to what happens when we got rid of BRCA1. We could show that this was also true in multiple ovarian cancer cell lines as well as non-ovarian cancer cell lines, that getting rid of CDK12 causes pretty profound sensitivities to veliparib. And one thing I want to point out to you is that if you're not familiar with, these are actually colony-forming assays. So what we do is throw a bunch of cells on the plate to get individual colonies to grow out and then count them. It's a very simple but tedious assay. But just notice that these are log scales here so that the effects that you're seeing are actually more profound than what you might initially think. Okay. So the summary for this part of the talk is that most ovarian cancer-associated CDK12 kinase domain mutations severely disable the kinase activity. And depletion of CDK12 disrupts HR repair, as well as some of these mutations also disrupt it and cause sensitivity to cross-linking agents, which happen to be what are actually used to treat ovarian cancer, at least up front, as well as to PARP inhibitors. And so our thought is that this is another way for ovarian cancers to acquire defects in homologous recombination separate from mutating BRCA1 and BRCA2. And in fact, if you go back and look at the TCGA database, what you see is that the tumors that have defects in CDK12 do not have apparent defects in BRCA1 or 2 or other homologous recombination genes. All right. So now we're going to switch gears, still sticking with ovarian cancer, and some of it will connect with what we already talked about, is going to DNA damage checkpoint signaling. And I always have to be careful now when I talk about checkpoints because everybody gets excited because they think it's immune checkpoints. But these are DNA damage checkpoints, and as Anya said, I've been studying these for 20 years, and when I first started, I really had no clue. I mean, I was actually hired at Mayo. I was a postdoc at Mayo and then got hired there as a faculty member with the directive to go study DNA damage, despite the fact I had never studied DNA damage ever. And so this is what happened. So this is sort of the 30,000-foot view of DNA damage checkpoint signaling at this point. And there's 2 structures that activate these 2 pathways. And this is really the simplified version. And on the one hand, on the left side, you can see double-strand breaks will activate the kinase ATM, ataxia-telangiectasia mutated, which regulates DNA repair, p53, and which in turn regulates cell death and or G1 arrest. Okay. If we go to the other side, we have extensive regions of single-stranded DNA. Those will activate this pathway that most people call the ATR check-1 signaling pathway. And that regulates progression through S phase as well as G2 arrest and DNA repair and the stability of the replication fork. And these 2 pathways talk to one another in a number of ways, one of which is that you can have this converted into a double-strand break by some sort of nucleases, and you can also have this converted into single-stranded DNA that will activate the ATR pathway via these nucleases that prepare the DNA for various repair pathways. Our interest has focused on this pathway over the years, and this is sort of a little bit more mechanistic view of how this pathway actually works. And probably the best stimulus for the pathway is to block DNA replication. And you can block DNA replication in many, many ways, one of which is causing disruption of the DNTP supply that is required for replication, and that frequently occurs when you treat patients with a variety of many different chemotherapy agents, anti-metabolite type chemotherapy agents. And it also happens when you have lesions on the DNA that block progression of the polymerase. So, and what happens when you block progression of the polymerase? There's a helicase out here somewhere, not shown on this diagram, that just keeps trundling along, unwrapping the DNA. So apparently helicases are kind of dumb. Hopefully not to say anything too bad about them, but they don't recognize that the polymerase is stopped. But actually that's a key point in how this pathway is activated. What then happens is these extensive regions of single-stranded DNA are coated by RPA, and DNA polymerase will also start laying down little tracts of DNA, double-stranded DNA. And that's a signal to recruit this 911 complex, which is right here. It's composed of Rad9, Hus1, and Rad1. And it actually forms a ring that looks like pCNA. Structurally, it's very similar to pCNA. when we first started studying this, we made that prediction that it was a ring based on some biochemical studies, and the crystallographers took about a decade to catch up and tell us, yeah, we were right. And it's loaded open. It's a clamp that can be open and loaded, and it's loaded by RAD17, another checkpoint gene, in complex with a bunch of RFC genes. And it's a complex very similar to what loads pCNA onto DNA replication, sites of DNA replication. Independently of that, these structures also recruit a complex called the ATR A-trip complex, and so we have two different complexes recruited to the DNA independently, and then the 911 complex plays a critical role in activating this ATR A-trip complex. Unlike pCNA, Rad9 has this long tail that hangs off the donut-shaped complex, and we identified a whole series of phosphorylation sites in it, and through much work eventually figured out that the C-terminal 1 is primarily important for binding to a protein called TOPBP1. TOPBP1 in turn has this activation domain that can reach over and activate ATR and thus set in motion activation of CHEK1. Now, this is not the only way that ATR is activated, but this is one of the classic ways. CHEK1 is another kinase, and once phosphorylated, ATR and CHEK1 phosphorylate a lot of different substrates within the cell, but primarily they regulate DNA repair, they regulate the S and G2 checkpoint, and they can stabilize stalled replication forks. And all those things working together protect cells from DNA damage. And in fact, tumor cells rarely ever lose this pathway, unlike the ATM pathway, which is frequently lost in tumor cells. Okay. So having figured out that, this sort of checkpoint activation scheme, we started asking questions about what else could this 911 complex do aside from activate checkpoints. And we got interested in base excision repair because supposedly it played some role in regulating base excision repair. And when we started thinking about those experiments, it's like, well, what kind of DNA damaging agent would we want to use if we want to study base excision repair? And there's lots of them that you could use, but we thought we might as well use something that's clinically relevant and use that. And so we picked 5-fluorouracil, and 5-fluorouracil is really complicated to talk about because of all its effects on cells. But I'm going to walk you through it briefly here. So here's 5-fluorouracil. And about 95+% of it ends up as FUTP and is incorporated into RNA. But that's really not, people don't think too much about that's how it plays a role in killing cells. And so this is sort of like the RNA pathway. It can also be, deoxyribose can also be added to 5-fluorouracil. And that it's called, for reasons that become apparent in a moment, that's also called floxuridine. And so this FUDR can be phosphorylated by thymidine kinase to create FDUMP, which is a really potent inhibitor of thymidylate synthase, which is important for producing DTTP so that you can synthesize DNA. And so if you inhibit this, you really screw up DNTP levels in the cell, plus you cause the accumulation of huge amounts of DUTP, which normally doesn't end up in DNA, but as I'll tell you later, does in this case. You also produce another active metabolite, FDUTP, which DNA polymerase has apparently recognized just fine and put it into the DNA. And so generally it's thought that this is how 5-fluorouracil kills tumor cells, but if you go back and look, it's not exactly clear that people have ever agreed on this, and there's RNA damage may also be part of how this works. And so I want to tell you a little bit more about 5-FU versus this other thing called floxuridine. 5-fluorouracil is one of the oldest drugs that we have for treating tumors. It's over half a century old. Despite that, it's still one of the most widely used anti-cancer drugs, primarily because it has activity against some very common cancers such as colon, breast, lung, and many upper aerodigestive tumors. Floxuridine, in contrast, actually is an FDA-approved drug, and many, many years ago, it's almost as old as 5-FU, and it has activity in colon, breast, ovarian, pancreatic, gastric cancers, although most of you have never heard of it, and even many, when you talk to medical oncologists, many of them have never even heard of it. It's actually approved for treatment of liver metastases of GI cancers, typically colon cancers, by continuous infusion into the hepatic artery. And the reason this it was an interesting drug for this application is because it causes extremely high local exposure to the liver, but because the liver metabolizes it, there's a very low systemic exposure. And so that would be a way to focus the treatment on the liver mets. But it's rarely used these days because people think that other drugs work equally well, plus the fact that you have to have continuous infusion is a bit of a pain, a big pain. Okay. So why am I telling you about all these? Well, when we started studying 5-FU and wanted to look to see the kind of base excision repair damage, et cetera, I'll tell you sort of the punchline here is we never saw 5-FU causing any damage that was repaired by base excision repair in multiple types of human cancer cells. And so we were really puzzled by this. But it turns out that if we looked at floxuridine, we could see lots of damage induced that was repaired by base excision repair. I just want to walk you through a little bit of some of the studies we did with these agents. And the first thing, or maybe not the first thing, but one of the first things we did is ask, well, do they both induce DNA damage? And what we found is that if we looked at gamma H2X phosphorylation, which is a marker of all kinds of DNA damage, we could see that 5-FU, floxuridine, and then as a control, hydroxyurea all induced nice foci, and exactly what you'd expect in a couple of different cell lines. When we looked at activation of the checkpoint signaling pathways, we could see that both phospho-CHEK1 was activated, so indicating the ATR pathway is activated. Some weak activation of the ATM pathway as evidenced by phospho-CHEK2. And so we had activation of those pathways. And we then asked, well, do those pathways affect the sensitivity of cells to floxuridine? So we depleted cells of ATM and or ATR and asked what happens when we treat the cells with floxuridine and 5-FU. And we saw that depletion of either one didn't affect sensitivity to 5-FU, which was already kind of weird because everything we've ever touched that damages DNA, when you get rid of ATR, you make the cells more sensitive to it. So that was an unusual result. But in contrast, when we looked at floxuridine knocking down ATM but not ATR, massively sensitized those cells to that agent. So now just to give you a focus in a little bit more on what happens when floxuridine is in cells, is what you see, as I told you earlier, F-dump inhibits thymidylate synthase. You get DNTP disruption, huge amounts of DUTP accumulate, as well as FDUTP. And you essentially end up with the cell, the DNA studded with and substituted with uracil and 5-fluorouracil. So it's pretty weird looking. And these lesions are substrates, at least in vitro, for a variety of uracil DNA glycosylases. And there are 4 known glycosylases shown here. And what they do is they come along and they clip out the base, leaving the phosphodiester backbone intact. So you have an abasic site, which is recognized by an enzyme like APE1, which causes a single-stranded nick at that point. And then a bunch of magic happens. Proteins like XRCC1 and poly-ADP ribose polymerase, which I mentioned earlier, recognize that, and you can feed this into one of 2 different repair pathways that either removes a single nucleotide or a few nucleotides and resynthesizes and fixes the damage. This is going on all the time in your cells because we have lots of damage that's repaired by base excision repair. And so, as I mentioned, all of these lesions, at least with in vitro substituted DNA and purified proteins, will remove the lesions from those synthetic substrates. And we were really interested in what glycosylase was important in cells. Forgot to mention here, to show that base excision repair was actually important, we knocked down XRCC1, so one of the key components. And we could see that knocking down XRCC1 sensitized to floxuridine very nicely, not surprisingly, didn't sensitize to 5-FU. Okay, now we asked which glycosylase was actually important. And we got actually surprised here. When we knocked down UNG, and there's 2 forms of UNG. There's a mitochondrial form of UNG and nuclear form of UNG. UNG2 is the nuclear form, and UNG1 is the mitochondrial form. Our siRNAs took out both of them. They're just differentially spliced genes. And what you could see is that massive sensitization when we got rid of UNG, but no sensitization when we got rid of TDG, SMUG1, or MBD4. And this was kind of surprising because everybody had basically ignored UNG as being the glycosylase that removes lesions induced by these agents. So we were a little concerned about that. So we actually went and did this experiment in a whole variety of cells and asked whether UNG was important. And in all the cells that you look at here, UNG was important in everything but this one. So clearly it plays a major role in removing these lesions in a variety of cell types. So just to summarize this part of the talk, floxuridine, but not 5-FU, induces toxic lesions that activate the EHR check1 signaling pathway that helps these cells survive that damage. And those lesions are also repaired by base excision repair, and that repair is initiated by the glycosylase UNG. So one of the questions we had as we went along is what regulates UNG2? And that's the nuclear form, and I'll tell you right now is that when we take cells and get rid of UNG and then put back UNG2, the nuclear form, it completely restores their defects. So it looks like that's the key one, not the mitochondrial version. Okay. So this is what UNG2 looks like. It's got mostly composed of a glycosylase domain, and it's got this N-terminal extension that has some a PCNA binding motif as well as some RPA binding motifs. And it also turns out it's phosphorylated, and that's been known for quite a while. And to sort of get some try to get something new on UNG, what we did was did some tandem affinity purification of UNG, and then asked what was associated with it. And I'm not going to show you that data. We found PCNA, we found RPA, just like you would expect. But we found lots of peptides that were derived from glycogen synthase kinase alpha and GSK3 beta. And that was kind of weird to have kinases hanging on to a protein through tandem affinity purification. But when we looked at the sites that had previously been characterized as phosphorylation sites, and they were actually thought to be CDK sites, we could see this, but actually this thing is really a GSK3 consensus site. And it looks a lot like what you might find in MYC and other ones where GSK3 phosphorylates the protein and regulates its stability. And so we thought, well, that's kind of interesting that we've got this kinase that's tightly associated with UNG. We have a motif that could be phosphorylated. Does GSK3 regulate UNG? Okay. And so this was research done by a student in the lab who just recently graduated, Carly Baer. And what she did was, let's see, this is where we took cells and, she just overexpressed UNG and either wild type or a mutation in either this site or this site, and showed that only the wild type really bound GSK3 very well. She then asked whether whether GSK3 regulates phosphorylation in the cell. So using a phosphospecific antibody, she could show that the wild type would react with this antibody, whereas a mutation of T60 or S64 did not. And I forgot to mention that GSK3 is a weird enzyme in that it has to have a priming site to phosphorylate its target site. So this is phosphorylated by something else. Once that's phosphorylated, GSK3 can phosphorylate a site 4 amino acids back. And when she mutated the priming site or the actual site we believe is a GSK site, we lose reactivity. And just to further shore that up, she actually converted this serine to a negatively charged amino acid and was able to partially restore phosphorylation of the target site. She also was able to show that inhibiting with a GSK3 inhibitor blocked these phosphorylations. And finally, she showed that by incubating purified GSK3 with UNG, we could phosphorylate GSK3, only the wild type, but not the T60A mutation. So that demonstrated that GSK3 is phosphorylating UNG. So then we needed we wanted to study what role this phosphorylation plays. So we created a cell line where we knocked out UNG. And then Carly put back a variety of different things back into the cells, either wild-type UNG, and this is a busy slide, I'm sorry, UNG with a mutation at the GSK3 phosphorylation site or the priming site. And these are all stable cell lines. And what I can show you here is that in a cell line that lacks UNG, not surprisingly, they're very sensitive to floxuridine. In the cells where we put back the wild type, we restored full resistance or reversed their sensitivity. Okay. So we wanted to ask whether phosphorylation affected catalytic activity of UNG. So Carly set up this assay where she produced PCR products that were either normal DNA or substituted with uracil, and then incubated them with cell lysates, and then just took the DNA, purified it, ran it on a gel, and saw if it was still there. And she could show that, in this case, she's using 5-fluorouracil substituted DNA, that if you added the lysate, you would get degradation. And that's just a time course. And when she studied these mutants, what she found is that the T60A and the S64A mutants, or cell lines harboring those mutations, had far less ability to remove uracils, at least in vitro, in these in vitro cell lysate assays. So it suggested or indicated that inability to be phosphorylated by GSK3 is associated with reduced catalytic activity of the enzyme, at least in vitro. So we asked, well, what about in cells? Turns out that measuring uracil or 5-FU accumulation in cells in DNA is tricky. So we did sort of a surrogate assay for this, and we actually used phospho-H2AX staining, which is an indicator of DNA damage. And so we took our cell lines and we incubated them with floxuridine for 24 hours, and you can see we got a whole lot of extra damage going on there. And then we washed the floxuridine away and said, okay, go fix the DNA. And if you look here, you can see that the cells with wild type substantially reversed the amount of damage, whereas the cells with the mutants didn't reverse that damage or repair that damage nearly as well. So there's clearly is a defect in repair in the cells as well. And so then we ask, well, what happens if we look at a ability to survive floxuridine treatment? And what we could see, these are just two separate clones. And what you can see is that these are T60A mutations and these are S64A mutations. And both of them have a defect in terms of sensitivity to floxuridine, so they're more sensitive to floxuridine, albeit not as sensitive as the null cells are. So clearly GSK3 plays a role in regulating in these cells. So just as a summary here, GSK3 phosphorylates on 3-adenine-60. Mutations of those sites cause defects in vitro and in vivo repair, and those are associated with sensitivity to floxuridine. Okay. So going back to what else floxuridine does is we started asking, well, are there any other DNA repair pathways that are important for floxuridine? And to make a long story short, here are the repair pathways that do not make any difference in terms of sensitivity. And to do this, we just took out key components of each repair pathway and said, does sensitivity change? But when we took out homologous recombination, in this case by depleting RAD51, but we could do BRCA1 or 2 and get the same result, we saw that they were very sensitive to floxuridine. So if you look here, we're taking out, let's see, UNG is here, and so you can see the sensitivity. If we knock down RAD51, we have about the same amount of sensitivity, and when we knock out both at the same time, we have really profound sensitivity. So to us, this was really good argument that both of these repair pathways are repairing these lesions, and that they probably are independent of one another, because you can see the massive increase when you take both out. So I want you to remember that for a second. We're going to come back to it later. So to figure out how floxuridine affects these cells, we started doing some studies, and the first thing we asked is, well, how does it affect transition through the cell cycle? So we just pulse cells with floxuridine, as well as some BRDU, and said, do they move through the cell S phase normally? And what you find is, at least with a lower concentration of floxuridine, is they go through the cell cycle just fine. You can see them marching through here, and they're actually, floxuridine didn't affect really the rate at which they went through S phase, although they did accumulate more in G1 24 hours later, which kind of caught our eye. And we also did the experiment when we knocked down UNG and saw exactly the same thing. Knocking down UNG didn't affect anything about how they moved through S phase, which was kind of surprising. So then we said, well, what happens if we look after the first S phase? So we allowed the cells to incubate in floxuridine for 24 hours, so to induce lots of lesions, and they actually will arrest in G1 reasonably well. And we did this with cells with control or UNG-depleted cells. And what you see is that they've arrested in G1 pretty well, and then we release them from the floxuridine arrest and say, how do you go through the cell cycle? And what you find is that they march through the cell cycle in the control cells pretty normally, whereas in the UNG-depleted cells, they have a profound defect in moving through S phase. Okay, so that suggested to us that there's some lesion induced by flox that's probably removed by UNG that's keeping these cells from going through S phase. And one idea is that if we had 5FU or uracil in the DNA, but presumably 5FU, that if that was in the template strand, when the cell came along trying to synthesize across that, that would be cause a problem. And so we actually collaborated with Marietta Lee at Medical College of New York, who has a really beautiful in vitro system to look at polymerase delta. And we created some templates where we could look at synthesis across 1 or 2 5FU lesions in these little synthetic substrates. And what we found, unfortunately, was there was absolutely no change in the ability of polymerase delta to negotiate these lesions. Okay, so that took care of that idea. So then we went back to the idea that homologous recombination was important for this agent, and said, could homologous recombination be somehow playing a role here? And so what we found is that when we treated cells, we took either control or undepleted cells. We treated them with floxuridine for 24 hours, and then we washed it off and looked to see what happened. And we could see that there was accumulation of lots of RAD51 foci, particularly in the undepleted cells that hung around for a long time. And we also saw the accumulation of RAD51 foci, indicating that homologous recombination type process is trying to occur. And so the question is, what are the lesions that are causing this homologous recombination problem? And it wasn't clear that it was going to be uracil or 5FU. And one of the things that we thought about is that it had been shown before that some glycosylases will actually create lesions that are more toxic than if the glycosylase never saw it. Okay, and I can give you an example right here, which we used as a control, this SMUG1 and this hydroxymethyldeoxyuridine. Okay, so if you give cells hydroxymethyldeoxyuridine and they have SMUG1, they're going to be sensitive with this curve. And this has been shown with multiple cell types with multiple investigators. If you take away SMUG1, now the cells are much more resistant to that lesion. So somehow when SMUG1 acts on those lesions, they become more toxic. And it's not exactly clear what happens. But we reasoned that if UNG wasn't removing the 5FU lesions, perhaps one of the other glycosylases was now creating more toxic lesions. And so we did experiments where we took away UNG plus each of the other individual uracil glycosylases, and then asked how do they affect the accumulation of RAD51 foci as a marker of homologous recombination. And so what we saw is that when we take away UNG, we get a lot more RAD51 foci. When we take away any of the other ones individually, nothing happens, not unexpected. But when we take away TDG, we can actually reduce this, and possibly a little bit with NBD4, but I don't think so. But TDG clearly looks like it's somehow creating these lesions that are engaging homologous recombination. And so that's our explanation for what's going on. So just to sum that up in a model that hopefully is fairly simple, if you put in 5FU lesions or uracil lesions, if UNG comes along and fixes that, everything's happy. The cell goes on and fixes and survives. If there's no UNG, we get an accumulation of these lesions. And TDG comes along, creates something. We don't know exactly what it is. that will then engage the homologous recombination pathway. If that fixes it, everything's good. But if that fails, we have death. So that's sort of a quick summary of what we think we found. Okay, so, and I just told you that, so I won't repeat that. So one of the things that caught our eye along the way, and this is where we get to the translational aspect, is that floxuridine, but not 5-FU, was engaging, the lesions were engaging base excision repair. And as I told you, poly-ADP ribose polymerase plays a key role in base excision repair. And so we asked a few more questions about what's the role of poly-ADP ribose polymerase in cells treated with this. And just to give you a little bit of background about poly-ADP ribose inhibitors, which are really quite exciting drugs in lots of tumors that have defects in homologous recombination. And that's because they're much more toxic for cells with HR defects than they are for normal cells. Okay? And because these HR defects occur in triple-negative breast cancer, ovarian cancer, prostate cancer, these are emerging as really interesting potential treatments for these tumors. And in fact, the first PARP inhibitor was approved for treating ovarian cancer in December 2014. And there's currently lots of massive clinical trials going on with a whole series of PARP inhibitors in different diseases and different combinations, trying to figure out how best to use these agents in the clinic. Okay. But back when we did these initial studies, PARP inhibitors were not yet approved, but there was a lot of excitement about them. And so what we did was, first of all, asked if we knock down PARP in cells, what happens? And we could show that we could sensitize cells to floxuridine, but not 5-FU. And then if we use PARP inhibitors, and here we're using two different ones, this is olaparib and this is veliparib, we could sensitize cells very nicely to floxuridine, but again, not 5-FU. And when we saw this really profound sensitization, we said, well, maybe this would be clinically interesting. So I went and found some colon cancer physicians and said, well, you know, floxuridine is used to treat colon cancer. Is this of any interest? And they said, does it work with 5-FU? And I said, no, it doesn't. Well, go find something else. Basically because they don't use this floxuridine anymore. So I went back to my office a bit dejected and started searching around, well, has floxuridine ever shown activity anywhere else? And it turned out that over 20 years ago, there had been a couple of clinical trials showing that it had activity in ovarian cancer. And the way they did it is they would actually just flood the peritoneal cavity with a bunch of floxuridine-containing solution, and that was pretty much the treatment, and it had activity. So I'm thinking, okay, there's this old drug nobody loves that has activity in ovarian cancer. Here's these new PARP inhibitors that everybody's excited about, and I can sensitize cells to floxuridine with PARP inhibitors. And would the ovarian cancer people perhaps be interested in this? So I went and talked to Lynn Hartman, one of our ovarian cancer, she retired recently, and said, would you be interested in this clinical trial? And she said, that sounds really interesting. So she and her colleagues, and a little bit on my part, we went through all the work to try to get a clinical trial set up, and eventually we did. And in the course of all this, there was, as with many drugs, they have this initial excitement, and then they have this trough where people aren't excited about them, and that occurred before it was even approved. In fact, some drug companies sold off their PARP inhibitors during this period. So we had to get through this trough to get this clinical trial approved. So it took quite a while. But anyway, we persevered. And so I'm jumping ahead of myself. But anyway, there is a clinical trial. Here's some of the other, I'll come back to it. There's some of the other data that supports this trial. So what we showed was that in a variety of cell lines, PARP inhibitors would sensitize to floxuridine. We took a couple of cells that were not immortalized, or that were immortalized but not transformed, and said, does the same thing happen? And no, PARP inhibitors do not sensitize. Here, this is ovarian surface epithelial cells immortalized by TERT, and here's some non-transformed fibroblast-type cell lines, and they were not sensitized to floxuridine by PARP inhibitors. And we did a couple of studies to ask, well, what kind of sequencing do we need to do with these drugs, at least in vitro? And we could show that what you really wanted to do was have the PARP inhibitor on during and after the exposure of the floxuridine. So that went into informing our clinical trial. And we also showed that what happens when you combine PARP inhibitor with floxuridine versus a whole lot of other drugs that are used to treat ovarian cancer. And these include, well, here's the floxuridine study. Here's topotecan, which is a second-line treatment for ovarian cancer, melphalan, second-line, cisplatin, which is the upfront treatment, doxorubicin, second-line, gemcitabine, etoposide. So for the few of them, we saw a little bit of sensitization, nothing like what we saw over here. As a control, we also threw in temozolomide, which induces lesions repaired by base excision repair. It's not used to treat ovarian cancer. And we saw profound sensitization, which is what we would expect. It's sort of the poster child for combining a chemotherapy agent with the PARP inhibitor. Unfortunately, it works so well that patients can't tolerate it because it cleans out their bone marrow. So this is not currently used. And then we also asked, what happens when you take away homologous recombination? How does that affect sensitivity? And we compared floxuridine plus PARP inhibitor to cisplatin plus PARP inhibitor in cells that either did or did not have BRCA1 in this case. You get the same results with BRCA2. And what you can see is if you take away BRCA1, you sensitize cells to cisplatin, but not to PARP inhibitor. And when you mix them together, not too much happens. In contrast, when you take away BRCA1 here, you sensitize cells to floxuridine. Likewise, you sensitize them to PARP inhibitor, and when you mix the two together, you have sort of Armageddon in terms of killing those cells. So they're incredibly sensitive to that combination if they're HR deficient. So summary is, for this section, is PARP inhibitor sensitized to flox better than other agents used to treat ovarian cancer? PARP inhibition during and after flox exposure increases cytotoxicity, and the flox plus PARP inhibitor combination is really toxic in HR deficient cells. So with that, we put together the rationale for the clinical trial that I got ahead of myself on. And the way this trial works is floxuridine is given by this IP therapy, and the veliparib, or ABT888, which is the PARP inhibitor, is given orally. And we're giving this to all patients because we know that the PARP inhibitor will sensitize to floxuridine, so and that should increase the toxicity of the floxuridine even in HR proficient tumors, but it might even have more activity in the HR deficient tumors. And this is a phase 1 clinical trial. It's currently underway in patients with metastatic epithelial ovarian cancer, run by Andrea Vonner Hendrickson. And I can tell you that I think we've put about 20-some patients on. We're increasing we go with the standard dose of floxuridine, and then we're ramping up the dose of PARP inhibitor. And there have been some sort of amazing patient responses on this. For example, one patient went through 33 cycles, and this is with recurrent ovarian cancer, so they've become cisplatin resistant or platinum resistant. One patient has gone through 33 cycles, stopped taking the drugs, and still has no evidence of disease. And there's a couple other patients who've had some very profound, lengthy remissions following this therapy. So it could potentially be interesting. We'll see. We've almost finished our dose escalation, and we're thinking about the phase 2 trial at this point. That was just and unless you're really interested in the clinical parameters of it, I'll skip that. But there's lots of PK studies and correlates that are going on. And this is just some of the patients that are the dose-limiting toxicities, which have, for the most part, been incredibly mild. In fact, one patient told Andrea, she said, "Well, I feel a little bloated after this, so I go running." So in that sense, it's been quite good. And you can see that the nausea, fatigue, neutropenia, and all those have been at pretty low levels so far. And like I said, some patients have done really well, and it seems well tolerated. And so with that, I'll just finish up and give my acknowledgments. We've got a really great group of people in the lab: Kathy Huntoon, Amelia, who did a lot of these studies, Purval, who did the CDK12 stuff, Carly, who did the GSK3 things, Bob Alver, who is an alumnus of here, recently joined the lab as a postdoc. And then I've got a lot of great collaborators: Scott Kaufman, Andrea Vonner Brooks and Joel Reed, other people who helped make this all happen, as well as Marietta Lee at New York Medical College, who helped with some of these studies. So happy to take any questions.
Thanks, Larry. I knew Larry when he was a graduate student, so you've come a long way, Larry. That was very interesting. The question, does it surprise you that the combination doesn't have the toxicity that the other combination had? I mean, how do you distinguish the difference? That it makes sense that if you're going to block a repair pathway and give second agents that, like PARP, that you would get a combined effect, but you would almost expect that there would be significant toxicity.
Right, and thanks for asking that, because I forgot to mention. So the reason the intraperitoneal therapy is useful in this case is because the systemic exposure is really low. So most of it is metabolized first pass by the liver. So we're actually doing pharmacokinetics on this, but the systemic exposure is really low, but the local exposure is very high. So we think that's why we're being able to avoid so much toxicity that's been found with other PARP inhibitor DNA damaging combinations.
So I might have missed it, but I'm in the back. But,
oh.
So does that mean you're not treating patients who have a pleural effusion?
Right.
Or disease elsewhere?
Right. They have to have confined to the peritoneal cavity.
Okay.
Which is where ovarian cancer initially typically is.
Right.
Yeah.
I actually have two questions. So CDK12, does it directly act on BRCA? Do you know how it stabilizes the protein? Is it indirect or is it directly
That's a good
connected?
question. So the RNA level goes down.
Okay.
So that's part of it. But whether that's the whole explanation is we're actually looking at that now.
And then in these double knockdowns where you deplete UNG and the other TS something, and you see the requirement for RAD51, do you know whether that is maybe a template switch mechanism so that it's post-replicative repair that's required there?
It's a good question. I don't know. Yeah, I mean, that would be a reasonable hypothesis, yes.
I have also a question about TDG. Is it actually able to excise out fluorouracil as well as UNG?
In vitro, yes.
In vitro, okay.
Yeah. In vitro, nobody's really looked.
So I did have another question. That is that given the fact that DNA repair damage or defective DNA repair is presumably a cause of a malignant transformation, your therapies are kind of accentuating that to the point of killing. Do you worry that you now potentially one of those side effects might be an induced second malignancies?
I mean, that's always the case. Anytime you treat someone with DNA damaging agents, are you going to get a second malignancy? I mean, my typical answer is we'd be lucky to find out. I mean, that would mean the patients would live long enough to get a second malignancy. So I think there's a reasonable chance that could happen, yes. But we don't. There are not a lot of options.
Given this disease, that's a risk that
you would be willing to take. Yes.
So in the sensitivity experiments, the floxuridine, have you selected cells for acquired resistance to floxuridine to see if the mechanisms are similar to de novo resistance?
We have not. Primarily because most of those, I've never done with floxuridine, but so many times when people select for resistance to metabolic agents like that, you end up with lots of things that block entry of the drug or metabolism, and I wasn't convinced that was going to be interesting. But we haven't done it.
And second part of the question, so there's a working hypothesis in the PARP inhibitor world that the drugs that trap PARP complex are more effective than drugs like veliparib. Do you see that in your model system? Have you looked at any others?
So that's a contentious issue.
They're certainly more myelosuppressive,
Yes. I mean, the question there really is yes, they will trap. And some of the drugs that don't trap, that purportedly don't trap as well, in some of the other studies where they've looked at it more carefully, they may trap just as well. They're just not, the assays don't detect it. My thought is that it really has to do with how good that drug is in terms of inhibiting. And the ones that inhibit really, really well do seem to trap pretty well. And they are more, correspondingly more toxic, et cetera. But we haven't looked at the trapping aspect of it at all.
Hi. So you excluded a number of DNA repair pathways that were not required for cellular resistance to floxuridine, and on your list was the Fanconi anemia pathway. And then you went on and said that, well, when we knocked down BRCA1 or RAD51, which now are officially also members of the Fanconi anemia pathway, you did see. So when you say key members that you knocked down, what were they?
We knocked down FANCD2.
Oh, wow.
Yeah.
And you did not see
Did not see a thing. Yeah.
Okay. Okay.
But we would see, and I mean, we did controls. We saw sensitivity to cisplatin and other cross-linking agents. But so this is different. Yeah, it's probably more a replication fork restart type effect that we're seeing, as opposed to a Fanconi anemia ICL repair pathway, which makes sense.
Does it? Well, FANCD2 is a key factor in replication fork restart and recovery, supposedly.
Yeah.
We can talk about it later. We'll meet. Okay. Okay.
Other questions?
All right. If not, then let's.
Thank you.